Well, good morning. It's still morning. My name is Cecilia Larocca. I'm a dermatologist by training, and I work in a multidisciplinary setting over at the Dana-Farber, seeing patients with both cutaneous T-cell lymphoma and cutaneous B-cell lymphoma. And by way of full disclosure, I am actively involved in research, not only clinical research, but also some really exciting stuff that sort of borders on translational and basic science that I believe Dr. O'Malley can give us a little bit more information on.
My name is Marianne Tawa, and I'm a nurse practitioner in cutaneous oncology for almost 2 decades now, working primarily with patients with cutaneous T and B-cell lymphomas. And my area of interest more recently has been in the clinical trials arena. When I started taking care of patients with cutaneous T-cell lymphomas, the chart of therapies was very narrow, and it was a very sad time for managing the disease. And over the years, seeing what has come through the pipeline that are now approved therapies has really sparked my interest in bringing that forward, because it's all about the care. I'm also involved with the Cutaneous Lymphoma Foundation. I serve on the board of directors.
And lastly, I'm John O'Malley. I am an assistant professor at Harvard Medical School in Dana-Farber. So I do clinic 1 time a month. So I recently switched to a new chapter in my life and took a job at Sanofi, which is a drug company. But in terms of conflicts of interest, what I do there has nothing to do with the oncology or CTCL drugs. So there's no aspect to that. But what I am there now is a senior clinical research director, help develop clinical trials, so have experience in that arena. And then prior to what I did, I had my own lab at Brigham and Women's Hospital, where I did research on early stage CTCL and prognostic markers. And it was with some sadness that I left the lab behind, but I am comfortable answering your questions about research today.
Great. So let's, why don't we start, Dr. Larocca? I think I'm going to toss this over to you to give a little overview of research, because I don't know about everyone in the room, but my immediate thought about research is treatments and clinical trials, but it's really much more than that. It's a much bigger umbrella. So to give us a little bit of context.
So I think just to take a big step back and sort of ask, well, what is research? Research is the pursuit of new knowledge in a systematic way and in an ethical way. So there's different phases or different types of research. First, I like to categorize it as basic research or clinical research. And when we talk about basic research, we talk about lab research. So in the labs with patient samples or cells or mice. And then we talk about clinical research. And clinical research basically is any type of research involving a human participant. So patients, people. Within clinical research, there are clinical trials. So clinical trial is a type of study in which a participant is assigned to an intervention. And that intervention is expected to have some sort of effect. And so the study is designed to measure that effect or effects on the participant. And that effect is expected to have some sort of measurable outcome. And that outcome has to be some sort of health-related question. Is it safety? Does it clear the skin? Does it make you itch less? Does it make you happier? So again, there's a number of different ways the question can be asked, but certainly has to be something that's predefined ahead of time, and it has to be able to be measured within the design of the study. So when we talk about clinical trials, and many of your providers might bring up clinical trials throughout the course of your care, the first sort of big picture sort of overview of how clinical trials are divided is in phases. And we've heard a little bit about that with Dr. Laura Pinter-Brown. She talked about phase 1 trials, phase 2 trials, and phase 3 trials. There's also something called a phase 4 trial, but that usually happens after a drug is approved. So in terms of phase 1 trial, these trials are first in human trial. So it's a trial for usually a drug. Let's just focus on therapeutic trials for now, that has never been tested before in an adult or in a human. And because it's never been tested before, the most important question that needs to be asked is this a safe drug? And what do these different doses of drug do on a patient? And so for most patients enrolled in a phase 1 study, we're not necessarily expecting that it might have a positive impact on your disease. But really, if a phase 1 study isn't done, then it's never going to get to a phase 2 or a phase 3 study where we are potentially looking for, is this drug going to have an effect on your cancer? So phase 2 trials, now we have some information about what doses should be used. We have some sense of understanding of how risky this drug is, what type of side effects you might experience. And they start looking at safety in a much more systematic way, using doses that we think perhaps might have a positive effect for the disease. So sometimes, even in a phase 2 study, we might be on a drug that could have an impact on your disease. And then a phase 3 study, which usually has a lot more patients, is designed to specifically ask the question, how does this drug affect the disease? And like we've heard before, and certainly now this is the new norm going forward for this disease, all phase 3 studies have what's called a comparator arm. So your doctor can't necessarily guarantee at the very beginning of when you sign up for a clinical trial which of the 2 drugs you will get, but usually it's 2 drugs or 2 types of interventions that are going to be compared. And in that type of trial, one of the exciting things is that's one of the first ways that perhaps you might get access to a potentially exciting medication that otherwise you would not have been able to use in the course of your disease. So with that, I'll probably stop and take any more questions.
Can you turn me on? Great, thank you. So I'm going to flip it a little bit and go to Dr. O'Malley. So can you talk a little bit about, basic science and research, and where does that fit in, and why is that part so important to this disease and to all of us as patients, and what happens in that arena in the lab, and what kind of things were you working on that actually have an impact on how we're living, how we're learning, and how we manage the disease?
Yeah, no, I would be happy to speak to that. And one of the things I needed to mention is, by the way, the views expressed are all of my own and nothing to do with the company. That's the one last thing. I'm getting used to this.
It's on the record.
Disclosures, exactly. But it's really a spectrum. So just as Dr. Larocca was talking about, where we have phase 1 through phase 4, and phase 1 starts with really a rationale that's built. So why would we even use this drug in this disease for this type of patient? And what is built on that is going all the way to mouse studies or even cell line studies, is to first identify what drives growth of cells, or if they have lymphoma cell lines, what drives the growth of the lymphoma cell lines. And the information that's gathered from working in the lab day to day and finding out, if we block this, does it kill the cell? If we don't block this, does it help it grow? Helps to build a story for potentially different antibodies or other things that are used in the lab to help kill the cells, to maybe think about, how do we transition this to actually to make it clinically relevant to our patients? And then there's this aspect of research. So that's really the basic, where what you think about in terms of the media of people sitting at the lab bench and doing work. Then there's translational work, which also is done at the bench side, but that is a separate kind of area of research, which is more of the area that I focused in, is where we have skin biopsy samples or blood samples, and patients are taking these different drugs, and some of these patients are getting better on the drugs, other patients are not. And so a question that we may ask is, why did it work in some of these patients and why didn't it work in these other patients? And so then you do studies on the tissues that you have available to you. And as you can imagine, probably as humans, we are very genetically different, a lot of different variations. And so you need a lot of samples to be able to study those, to really start to see signals of why one patient or a group of patients did better than the other. And then we start to see what different things are exciting and work in the lab, in the mouse studies, in maybe small numbers of patients. And so that's when the trial is built. What you have to really prove to the government agencies and to the boards of the different institutions that are going to be doing it is, or not necessarily prove, but at least have supportive data for why you're even doing what you're doing. And with the evolution of our understanding of these different diseases, how our own immune systems can fight these diseases, that's really kind of what sets the stage then for these clinical trials. And then you hope for everyone who has a disease, and if it's not going as well as planned with the therapies that we currently have available, is that these newer things down the line are coming in. So then specifically in my own lab, what we were interested in is, there's a subset of early stage patients who continue to do well. They may have rashes that come and go throughout their time. And then there's a subset of patients that unfortunately, for reasons that we are trying to learn to understand, progress from skin-only disease to blood disease, lymph node disease, and don't do as well. And so we are very lucky at Dana-Farber where we have a lot of clinical samples, and we started to study kind of how different aspects of those samples can give us indications of how patients are going to do over time. And so I think a key thing and by the way, the Cutaneous Lymphoma Foundation was very kind and were very instrumental in actually some of the findings that we had from our research, not only from my lab, but also in Dr. Tom Cupper and Dr. Rachel Clark's lab in our understanding. But I think to caveat, we have some exciting findings, but I think one of the things that we always have to do is when research is first produced, it's always important that no matter how exciting the finding is, that research is validated and verified throughout time, right? We don't want to hang our hats on something that then we start to test it. The important thing in science is that we always test the results that have been found in other places so that we can see how supported that data is and how long and how strong that data is. So even in clinical trials, after the trial is done, even if it shows effect, we and doctors and the companies that develop the drugs continue to monitor that drug, to continue to show that it's safe and to continue to show that it's effective. And so I think that's the important aspects for that whole spectrum of what I just discussed. From the preclinical side of things, basic is that finding continues to be validated by multiple groups and multiple different individuals as a drug, as the concept first goes, from the drug first going into patients all the way throughout all of the clinical trials, and then even after FDA approval, continuing to follow the safety of that drug. I won't get into my specific things today. We can do that later. I want Marianne to talk.
We can. Well, I have a lot more questions, but I'll just flip it to you, Marianne, to talk about. So, what happens if someone is interested in a clinical trial? So what kinds of things should you be looking for? What do you ask? What does that entail? And I don't know how many folks in the room have been involved in a clinical trial. Just a couple. Okay. So maybe for everybody else, like when would you consider and what does that look like?
Right. I was actually going to ask that question too. How many people had taken part or had a clinical trial ever been introduced as a potential concept for you? So I think, historically, a lot of patients think of clinical trials as a sort of a last-ditch effort, or I've tried and failed everything, now a clinical trial, and then after the clinical trial, now what? Clinical trials are not designed for end of treatment, end stage disease. Yes, indeed, there are some trials that are really for people that have tried and failed multiple therapies, but the clinical trials that are really available for patients in the CTCL community are the entire spectrum. So to have the clinical trial conversation does not necessarily have to be when you have kind of really advanced along and you're trying to slow down the pace of the disease. It could very well be an early stage disease. One of the clinical trials that we'll speak about later will kind of maybe touch on a little bit of what's out there in the environment is for early stage disease. So to have that conversation, if you're a 1B patient, so you've got multiple patches and plaques, and maybe you've been engaged in phototherapy, topical corticosteroids, there may be a trial for you. So it doesn't have to be after you've had a systemic treatment or 2 or 3 systemic treatments. So having the conversation early on is really probably a good, I'm interested in trials. So when something comes along and I'm in a like group, please have that discussion with me. So as a patient, the things that I instruct patients or bring up to patients is to take a look at sort of, what is the study? Am I like the other patients that the clinical trial is looking at? So it may be a disease variance. So clinical trials some of them may just be for patients with Sézary syndrome, so patients with peripheral blood involvement. Other trials may be for patients with large skin burns, patches, or plaques. So do I look like the other patients being studied in that group? If I do, maybe I'm interested in that trial. Am I looking to sort of delay moving on to the next step? So I've met with my clinician. I've tried and failed maybe 2 treatments. And I know on the horizon, there are some other treatments that might further suppress my immune system. I'm interested in maybe preserving my immune system. So, but before moving on to maybe a cell-killing chemotherapy, there's a clinical trial that maybe is novel. As Dr. Larocca mentioned, I might not have access to this drug because knowing how drugs make their way through the FDA, it may not appear in our clinic for 3 to 4 years. The experience and the opportunity is here now, so maybe I would think about it. So it's either sort of delaying treatment, or I sort of fit into the population of patients that are being looked at and studied in this clinical trial. So having that discussion and thinking about it is also, I think, important for the clinicians as you're seeing patients in clinic to always be thinking about them for a clinical trial. You know, you never want to pull the rug out if your therapy is working and you've achieved a close to a near remission, you're comfortable in your skin. You don't want to, sort of disrupt that and move on to a clinical trial. It's really when you're at a little bit of a roadblock, when you really need something new. The other things that patients need to think about for trials is, can I meet the logistics of the trial? How many miles away do I live from the center where the trial is being conducted? Am I okay with the more frequent monitoring? Because those of you that have been in the trial know that you're having more frequent biopsies. Oftentimes, you're having more frequent visits. For some people, it's very comforting to have more frequent visits because they feel like the surveillance of their disease is ramped up a little bit, and for them, that feels good. For other people, coming into the major medical centers more frequently is not what they want to do. So you need to really figure out how your life sort of fits in with the rigors of the trial. And imaging and all that. And then the unknowns, there's standard of care therapy. And as Dr. Pinter-Brown and Dr. Carter spoke about, the response rates of most of the treatments that we have for this disease, whether they be skin-directed or systemic, is somewhere between 30 and 40%. And now we have the clinical trials that, for the most part, are probably going to be within that same range. Am I willing to take that risk on the unknown when there's standard of care therapies? So, those are all, I think, important things that patients need to think about. And one other thing I wanted to mention from this morning is a lot of the trials, the rigors of the trial on the behalf of the pharmaceutical companies is much greater now than it was 10 years ago. We're looking at global responses. So we're not just looking for our skin to look better. We're looking for all the compartments, for the lymph nodes to be better, the blood compartment to be better, or the viscera if the solid organs. So there's a really high bar set for these trials. So for some patients, they may not meet the complete response rate, but they might be 49% improved. And for a lot of patients with a chronic disease associated with many symptoms, a 49% improvement is pretty significant. But the drug may not have the response rate that brings it up to a 60%, et cetera. So those are the sort of things I think patients should think about: is where are you in the spectrum of your disease? Is your current therapy working? And if not, take a step back. Is there something in the clinical trial arena, and can I make that fit into my life?
I just want to add one thing. So the other aspect is these trials, since it's not placebo controlled, you are at least getting a standard of care drug that the new investigational drug is being compared to. So it's regardless, you're going to get treatment. It just is, you may not know which treatment.
Right. And the other thing with clinical trials, so the only one with placebo, because clinical trials are ethical. I mean, there's an internal review board and it's ethically bound. The only trial that really I think I've seen in CTCL that had a placebo arm was Riziquimod, which is a topical trial. But the beauty of that is after 8 weeks, every patient on that trial rolled over onto study drug. I think that's the only time in my experience I've ever seen placebo used.
But I wanted to be clear that people have the chance, like no matter what, it is built into the concept that of course patients want to be treated.
Yeah.
Right. Very good. So I'm going to open it up to some questions. I always have questions, but do people in the room have questions about clinical trials, what happens in research, what we're trying to learn? Not yet, or you're just hungry? So, Dr. O'Malley, I have a question for you. What what's missing? So what are the things that we really are trying to learn and understand in the lab, right? That will have a positive impact on all of us at some juncture down the road? Because I think there's a lot of things we don't know, right? We don't know how to diagnose sometimes very easily. We can't tell under the microscope. So what are the kinds of things that we're trying to understand?
No, I think that's a great question. I'll try to keep it simple and to the point. But I kind of think of it in three major buckets. A lot of you have lived through the frustration of not getting the diagnosis up front. And then the question always remains is, if we can make that diagnosis up front, can we actually impact the disease long term? And that actually is still an open question. So there's that bucket of how do we get the diagnosis soon? I think the second bucket, at least in terms of how I think about it, are if we can get the diagnosis at an early stage, are there ways, one, that what I mentioned before is predict patients who are going to have worse outcomes? And if that answer is yes, are there ways that we can intervene to prevent that group from progressing? And those are still open questions for research. And then last, I would say, is how do we get above that ceiling of 30 to 40% overall response rates in our systemic medications for those patients who do have advanced disease? And there's a lot of research, and the people who are in the field are excellent researchers. So in terms of having a good group of people thinking about this disease, you all are very lucky, because there is a great group of people who think about this disease and are very passionate about it. But I think the more and more we understand about the genetics, the more and more we understand about drugs that are potentially already available, that we can quote unquote repurpose them to treat this disease that's more rare, that we don't have the numbers of lung cancer and other cancers. And then the more we can learn about how our body fights it and ways we can potentially regulate our own immune system with therapies, are all ways that we can potentially benefit all those impact those outcomes. And then in terms of getting the diagnosis earlier, we do have some newer tests, genetic sequencing types of tests that we can feel comfortable that this is helping us diagnose it earlier. And then we need to make sure that there's studies in place that we can answer those questions. Can we impact the disease if we get an earlier diagnosis? Can we impact the patients who we are able to potentially predict with a higher rate of them having progressive disease?
Good. Dr. Larocca has something to add.
And I just wanted to add to that, sort of kind of, a few years down the line, sort of some of the more clinical questions that we're trying to figure out is, well, I think one of the first things you learn is that you have many options. You saw Dr. Laura Pintos-Brown, diagram of all the drugs that are approved or at least used to treat cutaneous T-cell lymphoma. There are a lot of them out there, but the question is, which is the right drug for your cancer? And I think a lot of you probably feel like you've gone through this trial and error process of trying to figure out, well, which one's the one that's going to help for me? And to be honest, we don't have predictive markers. So we don't know by looking at your biopsy or by knowing your diagnosis, which of like the handful or 5 to 10 drugs out there is going to be the best one for you. So at this point, a lot of research needs to happen on trying to figure out what's the right approach for your cancer. And this is, I have to say, a shameless plug for the clinical trial that I'm running over at the Dana-Farber, where we've partnered with this brilliant engineer over at MIT who's developed a micro device. This micro device is smaller than the grain of rice. We directly implant it in the skin, and it releases up to 20 drugs, any 20 drugs that we've put into it. And then we rebiopsy the micro device, and we remove the skin, the tissue, the cancer tissue around the micro device, and we analyze it in the lab. We look about 3 days later, how did your cancer do to all these 20 drugs that are diffusing, kind of leaking out into your skin in different regions? So there's no area of overlap. And so our hope is now, with all this information, is there something that we can figure out about what your cancer is doing and how it responds to these drugs early on that might help us understand not only, immediately what drug might be best for you, but then combined with really powerful tools like genetics of your cancer and a couple of other more sophisticated imaging modalities. Could we now then understand, now that we have this wealth of data, kind of globally, how we can predict which drugs are better for which group of patients? And so I think we're sort of just at the beginning of, really getting to answer these really exciting questions. And so again, it's not only about trying to figure out, which new drug is going to be the best, but kind of which is the right drug for the patient. And like Dr. O'Malley said, at what point in their disease is it the right time to use that drug? So I think there's a lot of really exciting stuff out there. And we just need, patients to also kind of come along that journey with us. And it's not without sort of great respect and gratitude that, you volunteer your time and your skin to sort of help answer these questions.
I had a question. Is that what you're calling precision?
That's exactly
research?
it. It is precision oncology, precision medicine.
Who is entitled to have access to being involved in those studies?
Absolutely. So it is a clinical trial that's registered with the NIH. So clinicaltrials.gov. You go online, you can see all the details of the study, and any patient can enroll in the study, but you'd have to be seen at the center that's running the study. Because it's a small study, again, we first have to prove safety, that this device is safe to implant in the skin. We recruit patients out of one center. So at the moment, it's going to be conducted out of the Dana-Farber. With that said, with every clinical trial, one of the discussions that you have with your provider is, do I meet the inclusion criteria? Or is there something about my disease that would exclude me for participating? So for this particular trial, since we're implanting it in your skin, you have to have disease in the skin that's thick enough to sort of support having the device. And so it would be for patients who have plaque or tumor stage disease. So in particular, unfortunately, it wouldn't be as helpful for patients with Sézary syndrome, where it's just pink but thin skin lesions. So the name of the trial is quite long, but it's a pilot study of an implantable microdevice for cutaneous lesions of T-cell lymphoma.
I have a question. Who pays for all your clinical trials?
That's a great question. It's what we spend a lot of our time doing. So there's two flavors in which clinical trials come from. One is called the industry-sponsored trial. So this is where the pharmaceutical company has come up with the idea. They have come up with the financial planning, and they pay for the study. Then we have what's called an investigator-initiated trial, which is the physician, the doctor, the researcher has come up with the idea. And now they have to figure out how to get it funded. And they can do that in one of many ways. One is that they can go to a pharmaceutical company and say, hey, I think you might really like this idea. In fact, I want to use your drug in this clinical trial, and ask, does this drug for disease A work for CTCL, where it's not approved? And so you could have an investigator-initiated study that's funded by a pharmaceutical company, or you can apply for public funding. So your taxpayer dollars go to support the National Institutes of Health, the NIH, and you can apply for grants from the NIH and say, hey, this is really cool research. We need money. And so the government could give you money, or patient donations. So in fact, this clinical trial would not have gone off the ground if it wasn't for a patient who decided to sort of really help get it off the ground. I think one of the challenging things that come with doing research is that you need to kind of, just like Dr. O'Malley said, prove that you have the scientific rationale to do the research. And so to apply for grants, you have to have preliminary data. And so groups like the Cutaneous Lymphoma Foundation are critical for researchers, because they give you that seed funding to get it off the ground, to really prove to the big dogs, the NIH, that your research is worth doing.
In fact, Dr. O'Malley is a recipient of one of our Clarion grants, and look at him today. The other thing I wanted to mention, as far as who pays for it, sometimes your insurance company participates in the payment for the clinical trial too. So even for industry-sponsored trials, if you're receiving testing that is considered standard of care for your disease, so biopsies over time, perhaps getting CAT scans at 6-month intervals, or certain blood tests, your insurance company might very well be participating in the payment for the trial, as well as a pharmaceutical company, which is, overarching, paying for part-time for the nursing salary, or the clinical research assistant, or the supplies, et cetera. But it definitely is a mixed bag. But those are important questions to ask when somebody introduces the concept of a trial, is financially, what am I going to be responsible for? And Suzanne used the term financial toxicity earlier, and you're going to be hearing that term over and over and over, I think, in years to come, in caring for a rare disease is that there's a lot of financial burden. So you have to ask that question, and whether or not your insurance is going to be participating. Are you going to be left holding the bag for more co-pays, et cetera?
All good questions. Maybe we want to, I know we're probably just another few minutes before we head to lunch, and I'll open it up to the panel to talk a little bit about the clinical trials in general that are open in cutaneous lymphoma, kind of maybe a little bit about who they're covering, what they're targeting, and some of them that are in which phases, just as an overview. And just to let everybody know, we do our best to try to keep a listing of the open clinical trials on our website. We try to work with all of not the individual ones at individual institutions, because there's too many, but we try to keep the major ones that are doing new therapies in cutaneous lymphoma on the website, sort of as a first stop shop for you to go in and look, and you can look it up on the website and put in your state, and you can see what clinical trials are available in your area and all of that. You can always go to clinicaltrials.gov, but I even personally find that a little bit confusing. So we try to make it a little bit easier for you to start with us and then go from there. But maybe you can talk a little bit about what's going on, because I think it's pretty exciting.
So, like Marianne mentioned, there are stages there are therapies within clinical trials for different stages of cutaneous T-cell lymphoma. I think the three that I'll probably just mention today, because they tend to be the ones that are available at the largest number of centers. The first one is a phase 2 study called SOLAIRE. That's the SOLAR study. And it's testing a drug called cobomarsin, which is an IV drug. And that one's targeting early stage disease. So if you have stage, I believe 1B, so meaning you have more than 10% of your body covered with skin lesions, and have a little bit of blood disease and maybe very small in lymph node involvement, but have no heavy burden of blood disease or no Sézary syndrome, this particular trial can be potentially one that is available to you. This study, like most studies, do require that you fail at least one therapy. So again, if you are newly diagnosed and haven't been on a treatment for your lymphoma, you need to have at least tried one thing and failed that one thing before you can enroll in this trial. So that's an exciting one that's up and coming for our early stage patients. Two other clinical trials are for our more advanced stage patients. And I say advanced only because that includes our group of patients who have a heavier burden of disease in the blood, so Sézary syndrome patients. One of them is called the TELEMAC study, which is, I believe, a phase 2 study as well. This requires that you fail at least 2 therapies before you enroll in that. And again, it's also an infusion medication that we're particularly excited about. And then the last one that I'll mention that's also for more advanced stage patients, and again, advanced, I mean, because it includes all stages. Again, you must have failed at least one therapy, is a combination of an immune checkpoint inhibitor, so the immune activator pembrolizumab, a PD-1 inhibitor that you heard Dr. Laura Printer-Brown talk a lot about, in combination with interferon and injectable. So those are the 3 that I can certainly answer any other questions that you have specifically about them. But again, they span the disease spectrum.
Sorry. I'm looking for our home audience. They're sending me questions, and I'm trying to figure out what the questions are because I don't see them. I see the text.
So I think one thing I can mention is, every study builds on prior knowledge. So again, there's no phase 1 study that isn't done without some, animal model or cell line model or some data to suggest why it might work. For the phase 2 trials that we have, and even the phase 3 trials, sometimes we already have a signal of response from the phase 1 trial. And certainly we have a good understanding of some of the sort of more common side effects that one might experience. So even though you're signing up for a study, something that's going to generate new knowledge, there already is pre-existing knowledge to inform us of perhaps how it might work, if it might work, and what the side effects are. So again, it might not hold true when they do a larger study, but ask your doctor, well, why are you thinking about this particular study for me? What was the research from the earlier studies that makes you excited about it or interested about doing this therapy?
Yeah, I oftentimes suggest to patients that they ask the question when a clinical trial is brought up in conversation, what is it about this trial that made you think of me? You know, is there something that was found in the earlier phase of the trial? Is there something about the specifics of my disease or my responses to other therapies that would predict potential success for me on that clinical trial? I suggest that patients ask that question. So,
And I was just going to say, we did have a comment from one of our home viewers with regards to, maybe you can talk a little bit about this. So you go on a clinical trial and maybe it doesn't work for you. Then what might you be thinking of as a next step? So assuming that you've maybe tried a couple of other things, they didn't work, you went on a clinical trial, then what do you do? Are you done? What do you think?
So at any point in time, a patient can withdraw from a trial. And I've had patients withdraw from trials because, the wait time, because you're committing oftentimes to a whole day in your institute with, things move a little bit slower with collection of data, the amount of imaging, blood work, et cetera, when you're involved in clinical trials. So it's time off from work, which for some people that contributes greatly to their lifestyle, not being able to work or missing a day's work for clinical trials. So that might be the reason. The other is that perhaps you progress on the clinical trial and you would have to be removed from the trial, or you're just no longer interested. You just get clinical trial fatigue. You just need to stop. What happens next? It could be that you get your disease into a more steady state and you're comfortable. You sometimes could even return to a therapy that you had been on before that may actually work okay now that didn't before because you've sort of debulked or lessened the manifestations of the disease. So you might be taking a little bit of a break, which is good. For other patients, they might move on to another clinical trial because something new and exciting is kind of happening. Or they might move on to another standard of care therapy that was available, but maybe you didn't opt for that at that point in time because you left it in the wings for something that you might do in the future. So those would be a couple different scenarios.
Great. Oh, good. A question. Here we go.
How often does a patient, having completed a trial, go from the standard of care or the placebo, whatever you might want to call it, to the drug that's being tested? Because it turns out that it is more successful than the standard of care.
And Dr. O'Malley can certainly follow up on this. It actually all depends on the pharmaceutical company that's running the clinical trial. They might decide that ethically, because you had a response, even though the clinical trial has ended, but because you're doing so well, now they have an obligation to really continue to give you that medication, even though it's not approved and other patients don't have access to it. So if you're on a clinical trial and you had a good response and your doctor and you want to stay on that drug, some pharmaceutical companies, not all of them, might then actually continue to give that medication to you until it gets approval, at which point oftentimes there's a sort of delay phase, but once it gets approved, then you should be able to get that drug, and so could other patients. There are some times where the pharmaceutical company, either because they decide to no longer manufacture it, or if there's a specific reason, and again, I wouldn't know the details of that, perhaps Dr. Malley can comment on that, might not be able to continue to give you that medication, even though it was helpful. Again, overwhelmingly, the ethics are that if they have the medication on hand and you had a response, even though the clinical trial is over, you might be able to continue on that medication.
Yeah, and then the only other issue I wanted to comment on with clinical trials, the comparator arm. So you're all excited about the drug that's being studied, and the day of randomization comes and you wonder, am I going to get the study drug, or am I going to get what I could get prescribed by my physician, with their prescription pad right there. Many of the trials do permit, if you are on the comparator arm and your disease progresses along and you are not really responding to that standard of care drug, that's the comparator drug on the arm, or you have toxicities, you have side effects to such a magnitude that it is not in your best interest to stay on that drug, you would come off. And then many of the trials permit you to, quote, roll over to the study drug. So that's something to always be thinking about when a clinical trial gets introduced. I may not get that drug that I'm really fascinated with. I might get the standard of care drug. If so, what happens next?
Yeah, I mean, the only thing I'll add, Now, my experience is in the non-oncology space, but most of what happens are these open label extension trials, because it also benefits to find out the safety profile. So let's say the drug is working well. There's usually very strict criteria. You're not able to use other medications. But then at the end of the trial, if things are going well and the company is sponsoring to continue on it, then you go into the open label, which then also allows you to use other medications. It's not as strict anymore. So you're able to use other therapies in addition to the therapy that was working for you. And that benefits everybody, in my opinion, because there's more safety data then about how well people do over time. There's more longevity to it to see how long the effect is. So that's usually what happens, and that's termed the open label extension part to those trials.
Okay, so I have one more sort of question from our home audience, and then we'll go to lunch. But I think it's a good one. So as a patient, right, you get a diagnosis, a new diagnosis. It's new terminology. It's a new disease. You don't know anything about the treatments. So when do you start to ask about a clinical trial? Because sometimes if you are prescribed a certain standard of care, that could preclude you from participating in a future clinical trial. So can you maybe comment on how, as a patient, you might think about having that kind of conversation either up front or at some juncture as your disease changes and you're looking at different treatment modalities, so that you can have a better understanding before maybe you start a new treatment, what clinical trials might be available, so you don't eliminate yourself out of the box?
So that's a great question. I think it's actually a question that we're only now starting to think about. And the reason for that is that only recently did the FDA require that all drugs that go into a phase 3 trial be compared to a standard of care drug. At this moment, pretty much all clinical trials, and again, when I say all clinical trials, we're talking about a handful of trials, compare an experimental therapy to an HDAC inhibitor, which is either vorinostat or romidepsin. Now, vorinostat and romidepsin are, pretty attractive medication for many reasons. One, it works in all disease compartments, so it can work for skin, blood, and lymph node, although many would argue probably doesn't work well enough. And again, it's one of the, I would say, 5 most frequently used medications in CTCL. So again, ethically, if you're going to be randomized to either the intervention drug or romidepsin or vorinostat, that's good, right? You're going to get a drug that hopefully is effective. The problem that we're going to be running into now is if you've had vorinostat or romidepsin in the past, but you failed, so that drug wasn't effective for you, we can't ethically enroll you in that trial where you might be exposed to that same drug, romidepsin or vorinostat. So if you failed romidepsin and vorinostat, we can't in good conscience allow you to participate in the study because you might actually get assigned to the same drug that we know didn't work in you. So many of those, all of those trials then will prevent you from even enrolling in the trial if you failed vorinostat and romidepsin. So I think more recently in my own practice, I'm beginning to sort of reach for or prescribe vorinostat or romidepsin probably later on in my treatment algorithm, because I kind of want to keep it in my back pocket so that if you get enrolled into, or if we want to do a clinical trial, now we don't have to worry about your treatment past affecting your treatment future.
And that's only because we now have clinical trials and we've got new things coming into the pipeline. We didn't have a lot of options in the past and there weren't clinical trials. So, it's the good and the bad news, but I think it's things that for all of us to think about and to have conversations with our physicians.
The one catch to that is if it's a phase 2 trial where there's no comparator arm, everybody gets an experimental therapy, then that doesn't become an issue.
Right. Lots more we could chat about, but I know it's lunchtime and we're running a little bit late. So I'm going to turn it over to thank our wonderful panelists. And I'm sure if you're staying around, you can be available for any additional questions. And thank you for all of your work and your wonderful things that you've done and do for all of us.